was correct. Yeah, so thank you so much for the introduction. It's great to be here. It's a pleasure and it's an honor to be in this symposium. And today I'm gonna talk. About. I'm sorry. Part of the project that I developed with Martin Ramirez in Museo Argentino de Ciencias Naturales in my postdoc there. But I'll start talking about my PhD, the results of my PhD, part of my PhD in UFMG and in California Academy of Science, because it was back then when I started to get my interest in Gnaphosidae started. And I know that for the many people, Gnaphosidae might not look very interesting, like very appealing. They're just like brownish, blackish spiders running on the ground. But they have like incredible diversity, and they're not so big. They're like medium-sized spiders. But if you look close, if you look to the details, they have like a huge diversity of interesting characters to look. And so if you look at the claws, the spinnerets, Chelicerae and Sida, they have like a lot of interesting things that we can study and try to understand a little bit more about evolution, morphological evolution, and that reflects the taxonomical diversity, of course. So Gnaphosidae is the fourth species spider family, and they are all over the globe, except for Antarctica. And the main characters of Gnaphosidae, it's the modification of the spigots and the spinach itself. And so we have this huge modified anterior lateral spinnerets, and as Martin showed before, they have this huge piriform gland spigots, and usually the spigots are used as attachment disc. But as Martin was saying before, Gnaphosidae is a little bit modified. And so here, just comparison on your left, we have like other Leonita corinid, another family, and in red we're showing the piriform gland spigot, and on the right we have the piriform gland spigot of Gnaphosidae, and in blue in both figures we have the major emplate gland spigot. So we can see that in Gnaphosidae they're really like bigger and thicker than the major emplate gland spigot. So in Gnaphosidae, they are used to catch other spiders. So as Martin was saying, this paper by Jonas Wolf, they have this video showing how they use their spinning organs to catch the prey, and that's really cool. And during the presentation, every time you see those QR code, you can point to a cell phone and then redirect it to the sites for the paper in this case and for the video here. So you can see the Gnaphosidae catching prey with the stick silk that's spinned by the modified spigots. So this putative synapomorphy Gnaphosidae, this character that defines the group, were basically proposed by Plachnik when he in this paper actually he defined the families of Gnaphosidae, and he looked at the spinnerets, and he put some limits on the families and Gnaphosidae in general, and defines Gnaphosidae as having this modified spigot. And so this is the attempt to delimit the families in Gnaphosidae and delimit Gnaphosidae itself. And before that, there was like a lot of other papers, actually before and after that, dealing with generic limits in the family. And in many of these papers, they have mentions to super generic names like um, subfamilies, right? But those were not very clear, like the limits of those subfamilies. And they were basically like names that were used before, you know, by Eugene Simon or some modifications, of course, but like there were not a clear definition of what was Dracidine, Ecamine, Gnaphosine, and all other subfamilies or subfamilial groups. And the first attempt for doing that was Murphy's book. And in his book, he delimits groups of genera, 14 groups of genera. And he didn't treat the group as subfamilies, but it could be because many of the groups agree with the limits or traditional groups used as subfamily. And this was like really a really important step in the systematics and taxonomy. And this is a quote for the book where he said that he would expect that the book would serve for taxonomists to identify species or have like an idea if like this is a new genus or not, or which genus it belongs. And actually, I think that's like really important, and the job was really well done. And this is just like a quick look at the number of the increasing number of species in the 14 years before the publication of the book here in blue, and the 14 years after the publication of the book in orange here. And you can see like the increase in the number of species, in number of genera per year. So these are year intervals, and you can see that here in each year we have like increasing 1.2, about 1.2 genera per year. And after the book, like 2.2 genera described per year, like in average. So just like a quick look to see how the book was really important. And he said that the book is simply for identification purpose, and that character, how the character evolved were like more complex, and was not the idea of the book to show like evolution of characters and relationship of the groups itself. And he also said that this book should work as a graduate from passing from this interesting for some more serious study of the family. I think like kind of what happened to me when Adalberto proposed this, talking about Adalberto about this idea, was like really interesting. Like, what are the relationships between this group of genera? Are those monophyletic? Are the subfamilies or group of genera monophyletic? How they're related to each other? And actually how the Gnaphosidae are related to other families. The synapomorphy and then the monophyly of Gnaphosidae were never like really before that really thoroughly tested with phylogenetic methods. We have the paper by Martin Ramirez that's like on Nike spiders, and we can see that like maybe Gnaphosidae it's not monophyletic, but the sampling is not designing for tested that like thoroughly. So we decided to test the monophyletic Gnaphosidae and see the relationships of the group. So we sample outgroup taxa in the oblique median tapetum clade, or just like we see as a Gnaphozoid, extended Gnaphozoid clade. And with a denser sampling of this part of the tree here, that was like more the interest of the paper. And we sampled 71 genera of Gnaphosidae and also families and groups of proposed by Murphy from all continents and those spiders. And I have these 324 characters, and this is the person of the distribution of characters, and we analyze that in several ways. There's Bayesian and tidal weight analysis, and that's the tree like the results that we got. And we can see here first, that's those flattened gnaphosids. They are grouped together with other family. So you can see here the Vectus and Chloe, they're really, really supported clade. And if you look closer to the spinnerets, they're not so huge as we can see in the gnaphosids, but still like different in general. So in Chloe and Vectus, and here for platyoids. And so we thought, oh, maybe this is just like they are flattened. So this character is like giving too much weight to the flatness. But if we deactivate these characters, we got the same result. They're grouped based on other characters like genitalia and stuff. So we can see, so we consider that as trochanterids, we can see that still gnaphosids are polyphyletic, and we have this group here where we can see that some of the small gnaphosids also considered gnaphosids that have also, they don't have like a really huge piriform gland spigots compared to the template. And here also this Verita also, it's not so clear in a gnaphosid, but the relationship with the other families is also like not so certain. So if you thought that they're not gnaphosids, still gnaphosid looks like a paraphyletic, because in the middle we have protostomids as a branch of gnaphosid. And this is like a recurrent result found in other morphology related papers. So before, as I showed you, the paper by Martin Ramirez also shows something like that. And here the paper by Bruno Rodriguez and Christian Himes, also based on morphology with a big sample of protostomids, also show as a branch inside the gnaphosid. So if you treat that as an gnaphosid, we can have the synapomorphy as like this enlarged piriform gland spigot arising here at the ancestor of this group. And when you say large, you have like two characters. It's like bigger than the major arm plate and thicker than the major arm plate. So that are the only two traits, characters that support this group. There's no other, not even homoplasius character supporting that. And if you think about the evolution of this group, we have here the plasmomorphic state that we consider the base as long as the shaft, and with a clear division between the base and the shaft. So this is plasmomorphic for gnaphosид. And we have this modification here that's where the base is really long compared to the shaft, that we can see here in the what we call predominance and other gnaphosids. And also here it arises independently some other in other groups also. And if you look at protostomi, I have this classic, they're not only long, but they have these patches of seta associated to the piriform. So this is important. There are only once here, clearly. And we have this modification also when the base is not longer, the base and the shaft are not longer distinguishable. They have this continuous separate. We can see a little bit, but it's like kind of fused with both things. So it evolved here, but it was lost and evolved again a few other times. So we have this complex evolution of those traits and these characters related to spinnerets. And also, so we can also see some interesting things here, that the zelatine they appear as morpholike groups, and they're supported among other characters, also supported by the traditional characters like zelatine. They have this comb, printing comb of the fourth leg. And the airplane, they have this, if you look at here anteriorly in the chelicerae, this is one of the synapomorphy of the group. And this here we have a clade inside the airplane. We have a clade with you know, this, all the genera have this black, kind of black and white pattern in the abdomen. So this corresponds to basically to the Herpulus group in Murphy's book. And another thing that you can see here is that Gnaphosine, it's you know, should be the same as laranine. So we have this is synapomorphy of Gnaphosine, so the serrated keel here in the chelicerae, and then we have a modification of the serrated keel to this lamina in laramine. So another thing that you can see, that's Dracidine is also, it's not as it was delimited, but we can see a group that includes Dracids, and they have this notched trochanter in all legs. And this it's interesting character that arised a few other times here in the group. And also we can see like this transition from like this shallow indentation to like a more deep notched trochanter here. And we have this the new subfamily at the time, Ariscarvus leptodracini, that includes two of the Murphy's group, and they have like very that's Leptodrasus and Leptodracix group, and they have this very different and peculiar kinds of copulatory organs. So it's a very well supported clade in our analysis work, very well supported. And we have like a lot of other genera that are not you know, so stable, and some of them like Echimus and Anagraphs are their subfamily names, and but we cannot like clearly delimit those other subfamilies or the groups that were treated as subfamilies. So they're kind of still like unstable and not clear, the relationships, and or not monophyletic. And so this result was published in this paper in 2018. And so, but then we are interested in how molecules were you know, could tell about the evolution of composites, because the paper by Wheeler et al. Show like a really completely different topology, right? So you can see like the proteodomids, or at least proteodomine, are very, you know, distant related to Gnaphosid in general. So here we have Molecrini, that's we consider a proteodomid, and here the proteodomini, and here are the Gnaphosids. And if you take a look here closely, we have this huge mass. You know, so the molecular data is just like showing, you know, everything's paraphyletic. There's no clear relationship, but the supports are really, really small. So basically, the molecular data here is telling nothing. So we would need maybe to understand the Gnaphosids relationships, and we would need like to expand this more to Dionica, and expand also not only the taxon sampling, but also the gene sampling to understand molecular evolution. That. So if you look here, this is a recent published paper on the evolution of webs. They have like this phylogeny of spiders using genomic data, and you can see that Dionica here are very underrepresented in phylogenomics studies in general. So Dionica represents only 30% of all spider diversity, but like the sampling in those studies are really not representative of the real diversity. So we cannot tell much about the relationships inside Dionica. And, you know, that's like this huge diversity of, you know, morphological diversity, ecological diversity, and they're really like impressive and a really interesting group to understand, and very, very understudied. And so we don't know nothing, and we used to know nothing about the evolution of Dionica until 2017. That's the summary of the knowledge about the relationships are very not no at all. And if you look at like previous papers, so we have like Dionica here in red, and we can see that they are like most of the analyses appear monophyletic, depending of, so have morphological analysis, some traditional Sanger sequencing analysis here, combined Sanger sequencing and morphology, and here some genomic data, but also like the genomic data, as I told you before, was not very extensive sample for Dionica. And so we decided to explore more dynamic relationship using a genomic approach with user-conserved elements, but also including the traditional Sanger sequencing data and transcriptome data and morphological data. So we used the morphology the matrix of Martin Ramirez, including more taxon, and we analyzed this in several ways, all the matrix separate together in different approaches. And that's just like the conclusion of the relationship. So we can point here that we still have the same result as only with the Sanger sequence data. So predominant are really not closely related to Molecular and other Gnaphosids in general. So you can see predominant here, and here the other Gnaphosids. Then Gnaphosid and the Molecular, it's like a branch here inside Gnaphosid. So we thought we wanted to explore that a little bit more. We know that there are some things that can cause inference error, can cause error when we are estimating the species tree. And we know that the gene trees, they don't need to agree with the species tree. And in fact, most of the many gene trees will not be in complete agreement of the gene trees. But we expect that in general, the gene trees, most of the gene trees will agree with the species tree. But we have like a few gene trees that will not disagree. And here, but we have one case that we call the anomaly zone, where when the branches of the species tree are really short, that we can have this anomaly where the gene tree that agrees with the species tree, in this case, the C here, they would be very unlikely compared to the other one. So we have a lot of genes that don't agree with the species tree and just a few genes that agree with the species tree. And then if we infer or phylogeny with this data, we may get the wrong result. And so we thought if this could be an error caused by gene tree incongruence or a lot of incomplete linear sorting. And to do that, we tested with simulations. So we simulate alternative hypotheses, for example, predominant close related to Gnaphosium molecular, and we force this topology and estimate a lot of gene trees, simulate a lot of gene trees based on that. And then we simulate sequence based on these gene trees and concatenate, and then infer the species tree and compare the simulated with empirical. So we expect that this, when we simulate the data and analyze it, we expect that the gene tree would be the same as the tree that was used to generate that and not similar to the empirical tree. If we have a tree that's similar to the empirical tree, then we have a problem. So with this approach, we can have an idea and a probability of our data being in this like anomaly zone and being hard to analyze, and probably our data could lead to a wrong inference. And so our results suggest that actually it's very, very, very unlikely that the amount of incomplete linear sorting found in our data would lead to a wrong inference in relationship of the relationship of predominance, of predomining actually. So they're really, it could be really bad, you know. And we did other tests and also explore other possibilities, but it looks like that's what the molecular data and also the combined data with morphology that we use also show that. So the family should be established for this group, but like molecular should be kept in the Gnaphosid. So we have this evolution of the enlarged piriform gland spigots evolving several times. And I mean, this is not so weird because we have other kind of enlargement of piriform gland spigots in other dynamic groups. So all these stars here show families in which we may have some kind of enlargement of piriform gland spigot. And but the piriform spigot in Protodomids and millicrines and some Gnaphosids here, they have this condition that the elongation of the base, that's not common in other families. And if you look at the evolution here, it evolved at least 3 times here, and including like there are more Gnaphosids with this condition here that are not represented in the group. But if you look here, we can see that's, could be, okay, we know that this is a correct position. I mean, not correct, but at least our gene trees are not being led by confusion on incomplete linear sorting. But could be the case here, could be that these arise only once inside this group, and we have like an inference error causing a gene tree, a wrong species tree inference. And also, if you look this, we can see that Gnaphosids and lamponids are together. So Gnaphosids are paraphyletic in relation to lamponids. If you take a closer look at this part of the tree here, so we thought could be an inference error also caused by incomplete linear sorting. So we did the same test with the simulation. So we simulate here like those Gnaphosids with Gnaphosids and millicrines with a long base of the piriform spigot, and also Gnaphosid being monophyletic, but with a lot of incomplete linear sorting. And we did the same test, and we found that there is a chance that the amount of incomplete linear sorting are confusing our inference here in this part of the tree. So we cannot trust that much on that relationship that we're seeing here. So in order to understand that, we need to have increased taxon sampling and gene sampling for this and perform a thorough analysis. And that's an ongoing project that we're trying to do to expand this analysis for including more Gnaphosids and lamponids. But there are like a few interesting things. The sample is like small, but we can see that Zelatini and Ercolini are monophyletic, and Gnaphosini are, but like they include Amoxanus. This is like kind of weird, but like they are supported also by a few morphological characters related to not only the genitalia, but also other characters as well. And those results are published here. If you're interested, you can see the paper here, or there's like a thread on Twitter explaining some of the findings here. And so, in conclusion, like the phylogenetic the phylogenetic evolution of the Gnaphosids remains kind of elusive. This is an ongoing project. We still need more analysis on that and more data. And the clades are in general weakly supported by a few morphological characters that are homoplastic. So the morphology itself are not very informative, but also the genes that we have are also not very informative due to the amount of incomplete linear sorting with the molecular data. So we have these two kind of data that are like still not very informative about the evolution of Gnaphosids. So it's kind of hard. And also it's interesting like that we know this result that it tells us that we should look again at the piriform spigot morphology and development and to understand better that because their diversity might be like overlooked. You need to look at in details, and also how they use that would be very important to understand for us to understand better the homology of those characters. And with that, I'd like to thank you all the collaborators. Thank you all, and thank you for the invitation. Of course, like for Murphy for the great book. Without the book would be, my work would be like very hard to do. So And thank you all for the attention. And I'll be glad to take any questions.
Okay, thank you, Guilherme. Very interesting. It's clear that it's not that clear. Because of biology is still elusive, as you can see. We've got one question in chat already, which is about have you counted fossil genera, which about accounts for 6 genera and 25 species. Have you considered that?
No, I didn't consider the fossil. No, that's just like the living, the extant species.
Okay, and why?
But yeah, that's like a good some fossil records for Gnaphosidae also that I didn't consider here.
Okay. Any more questions, please? We still have time. Yeah, I know we have a number of people today who particularly study Gnaphosidae. Maybe you have questions about